students, welcome to NCERT Audiobook. Today we are going to start the 8th lesson of class 11th, Cell the Unit of Life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have, which a living thing has? The answer to this is the presence of the basic unit of life, the cell in the living organism. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others, like us, composed of many cells are called multicellular organisms. What is cell? Unicellular organisms are capable of the independent existence and performing the essential functions of life. Anything less than the complete structure of the cell does not ensure the independent living. Hence, cell is the fundamental structural and functional unit of the living organism. Anton van Leeuwenhoek was first saw the described alive cells. Robert Brown later discovered the nucleus, the invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cells. Cell theory. In 1838, Matthias Skachedian and German botanist examined a large number of the plants and observed that all plants are composed of the different kinds of the cells, which forms the tissue of the plant. At about the same time, Theodore Sechwan, 1839, a British zoologist, studied different types of the animal cells and reported that the cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on the plant tissues, that the presence of the cell wall is the unique characters of the plant cells. On the basis of this, Sechwan proposed the hypothesis that the bodies of the animals and plants are composed of cells and products of the cells. Sechdian and Sechwan together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow in 1855 first explained that the cells divide and new cells are formed from pre-existing cells. He modified the hypothesis of the Sechdian and Sechwan to give the cell theory a final shape. Cell theory is understood today is all living organisms are composed of cells and producers of cells. All cell arises from pre-existing cells. An overview of the cell. You have either observed cells, an onion peel and the human cheek under the cell of microscope. Let us recollect their structure. The onion cells, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains the chromosomes, which in turn contain the genetic material, DNA. Cells that have the membrane bound nuclei are called eukaryotic, whereas the cells that lack of membranes bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cells, semi-fluid matrix called cytoplasm occupies and the volume of the cell. The cytoplasm is the main arena of the cellular activities in both the plant and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Beside the nucleus, the eukaryotic cells have the other membrane-bound distinct structure called organelles, like the endoplasmic reticulum (ER), Golgi complex, lysosome, mitochondria, microbodies, and the vacuoles. In prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plants and mitochondria in all rough ER. Animal cells contain another non-membrane bound organelle called centrosome, which help in cell division. Cells differs greatly in size, shape and activities. For example, mycoplasma is the smallest cells are only 0.3 μm in length, while bacteria could be 3 to 5 μm. The largest isolated single cell is the egg of ostrich. Among multicellular organisms, human red cells are about 7 μm in diameter. Nerve cells are formed the longest cell, and cells also vary greatly in their shape. They may be disc-shaped, polygonal, columnar, cuboid, thread-like, and even irregular. The shape of the cell may vary with the functions they perform. Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pleuropneumonia-like organism. They are generally small and multiply more rapidly than the eukaryotic cells. They may be vary greatly in shape and size. The four basic shapes of the bacteria are bacillus, rod-like, coccus, spherical, vibrio, comma-shaped, and spirulina, spiral. The organization of the prokaryotic cell is fundamentally similar even though the prokaryotic exhibit a wide varieties of the shapes and functions at all. Prokaryotes have a cell wall surrounding the cell membrane except the mycoplasma. The fluid matrix filling the cell in the cytoplasm, there is a no well-defined nucleus. The genetic material is basically naked, not involved by the nuclear membrane. In addition to the genomic DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmid. The plasmid DNA confers certain unique phenotypic characteristics to such a bacteria. One such character is resistance to the antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor the bacterial transformation with the foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones of the eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotes have something unique in the form of nuclear inclusion. A specialized differentiation from a cell membrane called mesosomes is the characteristics of prokaryotes. They are essentially enfolding of cell membrane, cell envelope and its modifications. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, that is, the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs the distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of differences in the cell envelope and manner in which they are respond to the staining procedure developed by Gram. Those that take up a Gram stain and Gram positive and the other that is do not are called Gram negative bacteria. Glycocalyx differs in the composition and thickness among the different bacteria. It could be a loose sheath called the slimy layer in some, while in others it may be the thick and tough called the capsule. The cell wall determines the shape of the cells and provides the strong structural support to prevent the bacterium from bursting and collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. The membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosomes, which is formed by an extension of the plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and the lamellae. They help in the cell wall formation, DNA replication, and distribution of the daughter cells. They also help in the respiration secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extension into the cytoplasm called chromatophores which contain pigments. Bacterial cells may be motile and non-motile. If motile, they have a thin filamentous extension from their cell wall called flagella. Bacteria show a range of the number of the arrangements of flagella. Bacterium flagellum is composed of three parts: filaments, hook, and basal body. The filament is the longest portion of the extension from the cell's surface to the outside. Beside flagella, pili and the fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structure made up of special protein. The fimbriae are the smallest bristle-like fibers sprouting out of the cell in some bacteria. They are known to the help attached to the bacteria to rocks in stream and also to the host tissues. Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cells. They are about 15 nanometer by the 10 nanometer in size, are made up of two subunits 50S and 30S units, which when present together form 70S prokaryotic ribosome. Ribosomes are a site of protein synthesis. Several ribosomes may attach to the single mRNA and form a chain called the polyribosomic or polysomes. The ribosomes of the polysomes translate the mRNA into protein. Inclusion body. Reserve material in the prokaryotic cells are stored in the cytoplasm in the form of inclusion body. These are not bound by the any membrane system and lie free in the cytoplasm. Example, phosphate granules, cyanophycean granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. Eukaryotic cells. In eukaryotes includes all the protist, plants, animals, and fungi in eukaryotic cells. There is an extensive compartmentalization of the cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complexes, locomotory, and cytoskeleton structure. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as a former process cell walls, plastids and large central vacuoles, which are absent in animal cells. On the other hand, animal cells have a centrioles which are absent in almost all plant cells. Let us now look at individual cell organelles to understand their structure and function. Cell membrane. The detailed structure of the membrane was studied only after the advent and the electron microscope in 1950. Meanwhile, the chemical studies on the cell membrane, especially in human red blood cells (RBCs), enables to the scientists to deduce the possibles of the structure of plasma membrane. This study shows that the cell membrane is mainly composed of lipid and protein. The major lipid is phospholipid and the arrangement in the bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and hydrophobic tails towards the inner part. This ensures that the non-polar tail is the saturated hydrocarbons is protected from the aqueous environment. In addition to the phospholipid, membrane also contain the cholesterol. Latter, biochemically, invagination clearly revealed that the cell membrane is also possessed the protein and carbohydrate. The ratio of the protein and lipid varies considerably during the different cell type. In human being, the membrane of the eukaryotes has approximately 52% of protein and 40% of lipid. Depending on the age of extraction, membrane proteins can be classified as integral and peripheral. Peripheral proteins lie on the surface of the membrane while the integral proteins are partially or totally buried in the membrane. An improved model of the structured cell membrane was proposed by Singer and Nicholson in 1972, widely accepted as a fluid mosaic model. According to this, the quasi-fluid nature of lipid enables lateral movement of the protein within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of this membrane is also important from the point of the view of functions like cell growth, formation of intracellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many nucleoles can be moved briefly across the membrane without any requirement of energy, and this is called the passive transport. Neutral solutes may move across the membrane by the process of a simple diffusion along the concentration gradient, that is from the higher concentration to lower. Water may also move across this membrane from higher to lower concentration. Movement of the water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions and molecules are transported across the membrane against their concentration gradient, that is, from lower to higher concentration. Such a transport is an energy-dependent process in which ATP is utilized and is called active transport. The example, Na plus and K plus pump cell wall. As you may recall, the non-living rigid structure called the cell wall forms and the outer covering of the plasma membrane of fungi and plants. Cell wall not only gives shape to the cells and protects the cell from the mechanical damage and infection, it also helps in the cell-to-cell interaction and provides the barrier to the undesirable macromolecules. Algae have a cell wall made up of cellulose, galactines, mannose, and minerals like a calcium carbonate, while the other plants consist of the cellulose, hemicellulose, pectins, and proteins. The cell wall of the young plant cell is the primary wall, is capable of the growth, which is gradually diminished as well as matures, and secondary walls is formed on the inner side of the cell. The middle lamellae is the layer is mainly of calcium pectate which holds or glues the different neighboring cells together. The cell wall and the middle lamellae may be traversed by the plasmodesmata which connects the cytoplasm of neighboring cells. Endomembrane system. While each of the membrane organelles is distinct in terms of its structure and function, many of these are considered together as in the endomembrane system. Because of their function are coordinate, the endomembrane system include endoplasmic reticulum, Golgi complex, lysosome and vacuoles. Since the function of the mitochondria, chloroplast and the peroxisomes are not coordinate with the above components, these are not considered as a part of the endomembrane system. The endoplasmic reticulum ER. Electron microscopic study of the eukaryotic cells reveals the presence of the network of reticulum of the tiny tubule structures scattered in the cytoplasm that we call the endoplasmic reticulum. Hence, ER divides an intercellular spaces into the two distinct compartments, that is, lumeal inside the ER and extralumeal cytoplasm compartment. In ER often shows the ribosome attaches to the outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called rough endoplasmic reticulum RER. In absence of the ribosome, they accept the smooth are called as a appears the smooth are called as a smooth endoplasmic reticulum SER. RER is frequently observed in the cells actively involved in the protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site of the photosynthesis of lipid. In animal cell, lipid-like steroid hormones are synthesized in SER. Golgi apparatus. Camillo Golgi in 1898 first observed the densely stained reticular structure near to the nucleus. These were later named as the Golgi bodies after him. They consist of many flat, disc-shaped sacs and cisternae of the 0.5 micrometer to 1 micrometer diameter. These are stacked parallel to each other. Varied numbers of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near to the nucleus with the distinct convex cis or forming phase and concave trans or the maturing phase. The cis and trans phases of the organelles are entirely different but interconnected. The Golgi apparatus principally perform the function of packing materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packed in the form of vesicles form the ER and fuse with the cis phase of the Golgi apparatus and move towards the maturatic phase. This example why the Golgi apparatus remains in a closed association with the endoplasmic reticulum. A number of the proteins synthesized by ribosome on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus. Before they are released from its trans phase, Golgi apparatus is the important site of the formation of glycoprotein and glycolipids. Lysosomes. These are membrane-bound vesicle structures formed by this process by packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be the very rich in almost all types of the hydrolytic enzymes, optimally active at the acidic pH. These enzymes are capable of digesting the carbohydrate, protein, lipids, and nucleic acid. Vacuoles. The vacuoles is the membrane-bound space found in the cytoplasm. It contains the water, sap, exactly product of their other materials do not useful for the cells, and vacuoles is bound by the single membrane called tonoblast. In plant cells, the vacuoles are occupy the 90% of the cells volume of the cells. In plant, the tonoplasts facilitate the transport of the number of the ions and other material against the concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba of contractile vacuoles are important for osmoregulations and excretion. In many cells in the protist, food vacuoles are formed by the engulfing the food particles. Mitochondria. Mitochondria, unless specifically stains, are not easily visible under the microscope. The number of the mitochondria per cell is variable depending on the psychological activities of the cells. In terms of the shape and size also, considerable degree of the variable is observed. Typically, it is the sausage-shaped and the cylindricals having the diameter of 0.2 to 1 micrometer, length 1 to 4.1 micrometer. Each mitochondria is double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into the two aqueous compartments, that is the outer compartment and the inner compartment. The inner compartment is filled with the dense homologous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelles. The inner membrane forms the number of the infolding called cistae towards the matrix. The cistae increases the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of the aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called the powerhouses of the cells. Their matrix are also process the single circular DNA molecules. A few RNA molecules are ribosome and the compartment required for the synthesis of protein. The mitochondria divide by fission. Plastid. Plastid are found in all plant cells in the euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigment, thus imparting specific color in the plant. Based on the type of the pigment, plastids can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast containing chlorophyll and carotenoids pigment which is responsible for the trapping of light energy essential for the photosynthesis. In the chromoplast, fat soluble carotenoid pigments like keratin, xanthophylls and the others are present. This gives the part of the plant is yellow, orange and red color. The leucoplasts are the colorless plastid of the varied shapes and size with the stored nuclei. Amyloplasts store the carbohydrate and potato elysioplast are stored oils and fats, whereas the aleuroplast store proteins. Majority of the chloroplast of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped over spherical, discoid or even ribbon-like organelles, having variable length and width. Their numbers varies from 1 per cell of the chlamydomonas and green algae 20 to 40% cell in the mesophyll. Like mitochondria and chloroplast are also double membrane bound. Of the two, an inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. The number of the organized flattened membranous sac-like called as the thylakoids are present in the stroma. Thylakoids are arranged in stacks like of the piles of coins called grana or in intergranal thylakoids. In addition, there are flat membranous tubules called the stroma lamellae containing the thylakoid of the different grana. The membrane of the thylakoid encloses the space called the lumen. The stroma of the chloroplast contains enzyme required for the synthesis of carbohydrates and protein. It also contains the small double-stranded circular DNA molecules and the ribosomes. Chlorophyll pigments are the present in thylakoid. In ribosome of the chloroplast are smaller. than the cytoplasmic ribosomes. Ribosomes are the granular structure first observed under the electron microscope as the dense particles by George Paladin in 1953. They are composed of the ribonucleic acid, RNA, and proteins, and they are not surrounded by any membrane. The eukaryotic ribosomes is 80S, while the prokaryotic ribosome is 70S. Each ribosome has a two subunit, larger and smaller subunit. The two subunits of larger and smaller subunits of 80S ribosomes are 60S and 40S, while the 70S ribosomes are 50S and 30S. Here S is the Sjögren's unit, stands for the sedimentation coefficient. It is indirectly a measure to density of the size. Both 70S and 80S ribosome are composed of two subunit cytoskeleton. An elaborate network of the filamentous proteinous structures consisting of the macrotubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in the cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cells. Cilia and flagella. Cilia and flagella are hair-like outgrowth of the cell membrane. Cilia are small structures which work like auras, causing the movement of either of the cells surrounding the fluid. Flagella are comparatively longer and responsible for the cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that the eukaryotic flagella. The electron microscopic study of the cilium or the flagellum shows that they are covered with the plasma membrane. Their core are called exonema, possesses the number of the microtubules running parallel to the long axis. The exonema usually has 9 doublets, have radially arranged peripheral microtubules and pair of centrally located microtubules. Such an arrangement of the exonemal microtubules is referred to as the 9 plus 2 array. The central tubules are connected by the bridge and is also enclosed by the central sheath, which is connected to one of the tubules of each peripheral doublets by the radial spokes. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from the centrioles-like structure called the basal bodies. Centrosome and centrioles. Centrosomes is the organelles usually containing the two cylindrical structure called centrioles. They are surrounded by the amorphous pericentricular materials. Both the centrioles and the centrosomes lie perpendicular to each other, in which each has the organisms like the cartwheel. They are made up of 9 eventually spaced peripheral fibers of tubulin protein. Each of the peripheral fibrils is a triplet and adjacent the triplets are also linked. The central part of the proximal region of the centrioles is also proteinous and called the hub, which is connected with the tubules of the peripheral triplets by the radial spokes made up of protein. The centrioles of the basal bodies are cilia and flagella, the spindle fibers that give rise to the spindle apparatus during the cell division in animal cell. Nucleus. Nucleus is the cell organelle was described by the Robert Brown was early in 1831. Later, the material of the nucleus stained by the basic dyes was given the name of chromatin in by Fleming. The interface nucleus has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one of more special spherical bodies called the nuclei. Oli. Your electron microscopic has revealed that the nuclear envelope, which consists of the two parallel membranes, which is space between the 10 to 50 nanometer, called that the perinuclear space, from a barrier between the materials present inside the nucleus is that the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also the bears with the ribosomes on it. At the number of the places the nuclear envelope is developed, interrupted by minute pores which are formed by the fusion of two compartments. These nuclear pores are the place on both the direction between the nucleus and cytoplasm. Normally, there is only one nucleus per cell. Variations in the number of the nuclei are also frequently observed. Can you recollect the names of the organism that has a Lack of nucleus. Example, eukaryotes of many mammals and sieve tubes of the cells of the vascular plants. Would you consider their cells are living? The nuclear matrix or the nucleoplasm contains the nucleolus and chromatin. The nucleolus is spherical structure present in the nucleoplasm. It contains the nucleolus containing with the rest of the nucleoplasm as it is not a membrane bound structure. It is the site for the active ribosomal RNA synthesis. Larger and more numerous nuclei are present in the cells actively carrying out of protein synthesis. You may recall that the interphase nucleus has a loose and indistinct network of the nucleoprotein fibers called the chromatin. But during the different stages of the cell division, the cell shows the structural chromosomes in place of the nucleus. Chromatin contains the DNA and some basic proteins called histone, some non-histone protein and also RNA. A single human cell has approximately 2 centimeter long thread of DNA is distributed among its 46 chromosomes. You will study the details of DNA packaging in the form of chromosome in class 12th. Every chromosome, visible only in dividing cells, essentially has a primary concentric or the centromere on the side of which disc-shaped structure called the nitochores. Centrosomes hold the two chromatids and chromosome based on the position of the centromere. The chromosomes can be classified of four types. Metacentric chromosome has a middle centromere forming the two equal arms and the chromosome. The submetacentric chromosome has a centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming the one extremely short and one very long arm, whereas the telocentric chromosome has a terminal centromere. Sometimes a few chromosomes have non-staining secondary constriction at the constant location. This gives the appearance of a small fragment called the satellite. Microbodies. Many membrane-bound minute vesicles called the microbodies that contain various enzymes are present in the both plants and animal cells. Here the lesson is completed.